I'd like to welcome everyone to this session. It's my great pleasure to welcome Professor Geoffrey Gadd. Geoff is a Geo Microbiologist and mycologist with over 45 years of direct research experience of metal-mineral microbe interactions and their relevance to environmental processes and biotechnology. The Marjory Stephenson Prize is named after the Society's founding member and former president, Marjory Stephenson, and is awarded to an individual who has made exceptional contributions to the discipline of microbiology. I know you will all agree Geoff is a worthy winner, making notable contributions relating to fungal, metal, mineral transformations through multidisciplinary research at the interface of microbiology, mineralogy and geochemistry, and the establishment of Geo Microbiology as a recognized research area. He's going to talk to us today about fungal biomineralization. Welcome, Geoff.
Okay, thank you very much excuse me, for that introduction. I can see that the delights of Belfast have taken their toll on the audience, maybe, but it's a great pleasure to be here and a great honour to receive the Marjory Stephenson Prize lecture, which was established in 1953. Obviously, she was a great pioneer of microbiology and co-founder and second president of the Society of General Microbiology. It's wrongly put as the Microbiology Society on the website, tisk tisk, rewriting history, but I say that because the SGM was something part of my life since a PhD student, and I still can't get used to calling it MS or whatever. It's still SGM in my mind, as probably you know, some of us old guys, it still is. Coincidentally, I was also established in 1953, and whereupon my interaction with the world of microbiology, like all of us, commenced. And you can see there the young toddler blanketed by a crowd of microorganisms, microbiome, and all the rest of it. So I was asked to maybe just to summarize for interest, brief aspects of my career. Well, it's pretty simple. Department of Microbiology in Cardiff, 72 to 78, and then to Dundee, where I still am, 40 plus odd years later, and drastic degradation of features and all around bodily functions through the time. And, I've done all the other kind of stuff through the career in relation to the university, and some things I'm quite proud of is that when making the school, ensuring the future of microbiology, the standing of microbiology and establishment of the division of, which is now the Division of Molecular Microbiology, for example, in the School of Life Sciences, which is one of the biggest conglomerations of life scientists in Europe, actually. And there's a couple of shots of Dundee. You can see the famous rail bridge there that fell down about 100 years ago with loss of life. And the view from my side of the river, I live in a village on the other side of the Tay Bridge called Wormit, the first village in Scotland to get electric light bulb as a result of the Tay rail bridge being built. And that's one kind of view of Dundee from my side of the river. So metals, minerals and microbes have always been a focus and now I kind of realized in the last few years it's kind of under the heading of geomicrobiology, which really reflects the roles of microorganisms as geoactive agents. And we do work at and have done work on all kinds of scales and with all different kinds of organisms, but predominantly with fungi, although in the past we've had programs with cyanobacteria, microalgae, and for many years a program with sulfate-reducing bacteria, which again an important group of organisms, anaerobes, again, that like fungi in the aerobic environment encircle the planet and basically determine so many things about our life. Particular areas we're interested in, mechanisms of mineral dissolution, formation of novel biominerals, and their applications in, for example, bioremediation, biorecovery, and biodeterioration. And all the same mechanisms are involved in all these, so it's very nice. kind of easy in a way to see the applications, and I can't be saying too much about the latter today, but you can see various papers about biodeterioration in the built environment and of cultural heritage. So you can see kind of mineral in the title, Fungi and Mineralogy. So this is kind of under the heading of Geo Microbiology, if you like, the roles of a subset of Geo Microbiology, the roles of fungi in processes of environmental and geochemical, geological importance. And some of those areas there are what are involved: mineral dissolution, soil formation, structure and development, element cycling through decomposition, but also through rock and mineral transformations. And all the things that the organisms do, metal transformations are an integral part. And transformations of metals can be simply thought of as they either cause solubilization, increase in mobility, or somehow immobilization. So there's always this kind of balance. And amongst the immobilization mechanisms is biomineralization. Now, as you probably already know, fungi, the predominant, most fungi have filamentous growth pattern, organic nutrition. They rely on extracellular enzymes and products, particularly to break down larger organic molecules. And symbioses are very important with phototrophs, mycorrhizas, and lichens, which determine plant nutrition. 80% of plant genera are mycorrhizal, and rock and mineral transformations. And they must not be forgotten in all this topic. So the growth form and nutrition are the basis of the activities. If you look at a simple fungal model of interaction with rocks, minerals, and metal-containing substrates, quite simple is solubilization of the components, the metals, and they can then interact with environmental ligands and reprecipitate, as for example phosphates or carbonates or things like this. Join an environmental pool, but they also can interact with organisms, be taken up by transport mechanisms, or interact to form minerals, or form minerals outside in the environment. Solubilization processes generally involve organic acids, but there's also siderophores for iron, for example, protons and things like this. And of the immobilization mechanisms, biomineralization is a key part, and this is what I'm going to focus on. So biomineralization is the production of minerals by living organisms, and it occurs in all domains of life: bacteria, archaea, and eukarya. And it includes complex mechanisms under direct genetic control and uncontrolled mechanisms where the organisms are just altering their external environment through metabolism or just their structure, and which results in biomineralization. So you can talk about biologically controlled biomineralization and biologically induced biomineralization. And the biologically controlled mineralization is quite simple. There's one prokaryotic example, the magnetosomes in magnetotactic bacteria, which are a prokaryotic organelle. And then you might have, see things like this in alga, calcite deposition in Emiliana, coccolithophore, silica in a diatom, and then going up, barnacles, for example, crabs, lobsters, and then, of course, in our own situation, advanced biomineralization, skeleton, and various features, for various purposes. In this case, for example, teeth, for lying through. I can't resist saying that. And so, really, we're talking about in this context, in fungi, biologically induced biomineralization, which is quite peculiar to me, because why are fungi the only group that apparently haven't got any controlled biomineralization, whereas everything else has? But it could be we just don't know enough yet. So redox reactions, excreted metabolites, other species can cause this, and also the metal binding properties of cell walls, EPS, etc., can act as nucleation sites for minerals. And fungal mineral examples include carbonates, phosphates, oxides, and oxalates, particularly oxalates for fungi. And also elemental forms can be produced by reduction, such as selenium, tellurium, elemental silver. And this is an old paper we published in 1991 as part of studies on reclamation of silver from photographic wastes. you might know that there used to be these things called cameras that had films, and the basis of the black and white films was deposition of elemental silver. And of course, this generated quite a lot of waste. And, there are some silver nanoparticles, which we didn't think to call them nanoparticles at the time, but of course, you might know about nanosilver with its antibacterial and products. And I was a bit miffed about this, not thinking of this, to call it this. But of course, nanosilver has actually been in use for 100 years or more, because you can make it chemically, and it's been used for all kinds of things. There's some nanoparticles of elemental selenium around a yeast. And some of the processes there are analogous to making minerals. So a lot of fungal minerals, all kinds of contexts. There's a few here, some uranium phosphate minerals, calcium oxalate, rhodochrosite, which is a manganese oxalate, lindbergite, which is magnesium carbonate, and lindbergite, which is a manganese oxalate, some biomineralized organisms, deteriorating concrete, for example, and calcite, calcium carbonate, which, of course, is limestone or dolomite, depending on the... And so biomineralization, if you look at some aspects, maybe to do with a terrestrial environment, some work we've done with uranium, with depleted uranium, and there's some depleted uranium in soil from one of our sampling sites, depleted uranium missile testing range. There's 2 in Britain, and we've been testing of many years. The sea off the Lake District and southwest Scotland, there's tons and tons of depleted uranium on the seabed. And you can see yellowness, and the yellowness implies deterioration because it reflects the yellow uranal ion. And there you can see a close-up of these so-called hotspots. And this is a Hymenocyphus. It's a mycorrhizal fungus, but you can see it makes these strands. They're actually translocating uranal ion from the depleted uranium and can biomineralize and form these fantastic coralloid sheaths. And of course, they're dead at that point, and massive amounts of uranium accumulated. And a lot of work went into making the model, finding out what the minerals were. And it turns out that they're kind of uranium phosphate minerals of the meta-autunite group for geologists, chernikovite, uramphite, for example. There's well-known minerals to geologists, but this was the first time, a microbially mediated formation of such minerals. And bizarrely, these minerals are more stable than the depleted uranium itself. So if you're getting these processes in the environment, they're locking up the uranium and preventing it getting out and affecting other organisms, or even into water and affecting humans. And similarly with lead, we had quite an interest in lead. You always associate lead with some kind of toxicity, but also some kind of inertness. It's not essential biologically. And but when I used to be out walking, and if you see lead, which is kind of everywhere, you always see it coated with biofilms and traces of life. And sure enough, you can grow organisms on it, and things happen. So these are some minerals formed. You can see on the surface of metallic lead, you can see the hyphae and all kinds of minerals. And of course, we usually thought that these were just some kinds of oxides or carbonates or something, because metals oxidize in air. But when we examined, using all the usual mineralogical techniques, found that these minerals were pyromorphite, which is a kind of lead phosphate. Again, so phosphate's important. And again, this pyromorphite is the most stable lead mineral that occurs in the Earth's surface. And it's actually been used to remediate lead-contaminated soil just by addition of a phosphate source. Some of these soil people and geological people thought that this was just a purely abiotic reaction. You add a cheap source of phosphate, bone meal or rock phosphate or something like that, and the lead will precipitate and lock it up, and then you can use the land for something. But actually, this shows that organisms are involved, and just like phosphate fertilizers for plants, organisms are releasing the phosphate, which interacts with the lead. And we got some, good publicity about this. Conversely, as a sidetrack, we also have got fungal organisms that can actually make pyromorphite as well in some circumstances. Revegetation and reclamation, also biomineralization. This is some work with Thai friends in Chulalongkorn University in Bangkok. This is Padang zinc smelter. At the time it was one of the largest zinc smelter in Southeast Asia. And these are some settling ponds and things like this around the place. Very toxic. I was walking on the edge of one once, what I thought was a stable crust, and then went through the crust up to my knees in toxic waste, which, of course, by the time that everybody stopped laughing and I was hosed down, the metals interacted with my shoes, actually, and they were permanently stained. But anyway, this plant is everywhere, Lindenbergia, and one thing that we're interested in is why is it surviving, how is it surviving in such a place? And of course, it's down to the mycorrhizal fungi. It's mycorrhizal, and if you knock out the mycorrhizas, it can't grow in the presence of metals, and et cetera, et cetera. And you can get this biomineralized sheath around the roots that you can see here. There's some of the mycorrhiza. And the zinc is bound up, as well as other metals, as oxalates, carbonates, phosphates, and things like this. If we come closer to home, this is some work of Marina Femina, who I'll mention later on. Again, this was a BBRCC project on mycorrhizal fungi and reclamation of metal-contaminated soils. There's a Scots pine and zinc phosphate microcosm. Zinc phosphate, incidentally, forms in zinc-polluted soil, and it's a mineral. It's called hopite. And you can see some of the hyphae, and as we close in, you can see the organisms are altering the geochemical attributes of that environment. You get closer in, and there you can see so EPS, which forms a fantastic matrix for geochemical reactions, and you can see the crystal formation, some kind of zinc crystals, possibly oxalate, forming and dramatically altering and immobilizing the metal, and of course reducing or preventing translocation to shoots. Now, so that's outside. Now, interested in exploring these mechanisms for not only for bioremediation but for biorecovery. And as I mentioned, biomineralization is a result of metabolic activity, ligand and metabolite release. And a couple of areas that we've been very interested in is urea degradation that leads to carbonate release and precipitation of minerals with metals. Degradation of organic or inorganic phosphates that releases free inorganic phosphate that again can make metal phosphates, and organic acids, particularly oxalate, again, which can make all these different kinds of biomineral products. And mostly carbonates and oxalates, I'll be able to give some examples. Also, you can have reduction mechanisms, particularly with metalloid oxyanions, to produce elemental forms of selenium and tellurium, for example. And more about that in a minute. And so a very big area at the moment is metal biorecovery, recovery of essential elements, because environmental technologies and applications for clean and efficient energy production and usage rely on a range of raw materials, of which metals are of fundamental and strategic importance. And these can be called critical elements or e-tech elements, all these kind of phrases. And they're all the elements that are all in your computers and mobile phones, washing machines, microphones as well, microwaves. And you're used to these piles of electronic waste. You can see, TVs and phones. And at present, there's inadequate recycling and reclamation. A lot of it just goes to countries like India, China, and Africa, where people are working for a pittance in filthy conditions to pick apart whatever they can, which again will come back to us in our new iPhones and computers, and there's still a lot of waste. So a ton of mobile phones contains more gold than a ton of the best gold ore that's mined, for example. But there's a problem in recycling phones. I mean, nobody really does it to a great extent, but everybody knows about batteries. That's in everybody's mind from primary schools upwards. There are some smartphone elements, just to illustrate. It can be 30, 40, 50, 70, depending on the analyses. Gold and copper, obviously, lots of these elements, including a lot of rare earths. And you can see here just some of the important ones and where they're important. For example, important to environmental technologies. I mean, for example, selenium and tellurium are all solar cells. Cobalt is in the batteries for electric cars, and if you look at the numbers of electric cars postulated and the amounts of cobalt in the world are available, they just don't match up. So there's a problem about recycling reclamation, and particularly since EU has mentioned problem for the EU because, and I'm including UK in that, we import all these things. You know, we haven't got any supplies of our own. Many rare earths, they're only in economic quantities in 3 countries, South Africa, Brazil and China, who already have them on conservation lists, and China is being very clever, as usual. It's also buying up rare earth stocks from other countries so that they're going to have their computers and mobile phones when we're all sort of scraping around back to pencils and paper or something like this. And anyway, so just some examples of this. So selenium and tellurium reduction and immobilization. Very simple, very wide property in fungi, but also bacteria. And elemental selenium is red, tellurium is black, and you can see a fungus there, and precipitation red and black from selenite. But what is important and interesting is that the selenium and tellurium comes out as nanoparticles, and reactive supernatants can do this. And you can see here, you know, this controlled supernatant after growth of a fungus, and then you just add selenite or something, and red or a tellurite. And there's some of the nanoparticles you can see of selenium, little aggregate there, nice spheres, and nanorods in the case of tellurium. You know, not all nanoparticles, they have all kinds of shapes. They're not just little spheres as you kind of imagine. And so a mechanism for not only, say, remediation, but also recovery, in a useful form, nanoscale products, because nano selenium is what is used in solar cells and things like this. And metal oxalates are another precipitant that we come across as part of projects to look at leaching of cobalt and recovery of cobalt from resources. Aspergillus niger, for example, very effective. You know, you can get 90% removal from supernatants precipitation as oxalates, and also rare earths. There's a lanthanum oxalate, you know, big sort of massive fern-like crystals that you can see, and very nice to see, actually. And metal carbonate precipitation that I'll mention a bit later on. This is very interesting. It's already used for self-healing concrete, the bacterial stabilization of soil, where you're getting organisms to make carbonates that cement things together, but little used in context of recovery. And it's just where you have a urease-positive organism that breaks down urea into ammonium and carbonate, and the ammonium can be used as a nitrogen source. And of course, the carbonate will precipitate with any metals in the vicinity in the external matrix or associated with the biomass, for example. Now, of course, you're usually thinking of all these things of other organisms as well, and bacteria, and this is just an example of a work package from the COG3 project with many people, including John Lloyd from Manchester and Barry Johnson from Bangor, who worked on bacterial components, SRB and leaching, and Richard Harrington in Natural History Museum and other colleagues, mineralogists and geologists, where we have an integrated approach. So there was oxidative bioleaching, reductive bioleaching by acidophilic and metal-reducing bacteria. We looked at fungal leaching. Fungal leaching could do laterites, for example, but not sulfidic ores, which obviously acidithiobacilli and things like that can do extremely well. And then when you get the pregnant leach solution loaded up with the released metals, various kinds of treatments such as oxalate and carbonate, and also sulfate-reducing bacteria sulfite production. And, this was a very nice project, went on 4 or 5 years, and basically whatever we looked at, the combination of all these mechanisms, we could get all the cobalt out. A vast proportion of cobalt that we get comes from the Democratic Republic of Congo, and depending on the state of any civil war or whatever, the price goes up, and so of course you've got to find other additional sources and reclaim from existing sources. So back to biorecovery with carbonates. So quite simple. You have the carbonate, supernatant. You can recover almost anything, and from mixtures and all the kinds of things. But one intriguing thing that we found some years ago was that they were coming out many times as nanoparticles, and there you can see some copper carbonate nanoparticles there. And so immediately he's thinking, well, why are they coming out as nanoparticles? Because you'd expect them just to look like normal chemical carbonates that you might just synthesize from chemicals. And of course, amino acids and proteins seem to play a big role. You can see here the organisms naturally secrete a whole variety of amino acids. Glutamic acid is there as just an example. And of course, a lot of proteins, both naturally and also there's autolysis going on as organisms grow. And you can see there's some commercial copper carbonate, and there's some chemically synthesized copper carbonate, just by mixing, I suppose, sodium carbonate and copper chloride, something like that. And so you can see these very small nanoparticles that happen, about 30 nanometers or less. So this is one aspect to try and find out. There's some X-ray diffraction, and you can see obviously the spikes, they're characteristic of a given mineral, malachite in this case, this copper carbonate, and these are the characteristic patterns, and then it goes to this amorphous, and this is indicative of very small things that the X-ray diffraction can't work, and so smaller particle sizes and different mineral morphologies, and so the amino acids are changing the stability of the crystal precursors and inhibiting mineral growth. Then further, proteins that are secreted by Encrasa, and we used a lot of proteomic studies in this. And if you look at the proteins, loads of them were associated with the nanoparticles and removed from solution with nanoparticle formation. And some extremely strong association. So which proteins are involved? And Feishua Liu in the lab is a genius with this, and of the proteins, this triose phosphate isomerase was one that showed a particularly high affinity to bind with the copper carbonate nanoparticles, and you could synthesize the nanoparticles just with the TPI, in a test tube. And there's a lot of protein conformational changes, and the proteins were providing nucleation sites for mineral growth. Oh, and that's supposed to go, that video. Yeah, some confocal microscope. You can see the red, and you can see the green is the copper nanoparticles in that conglomeration there. And this attraction with the protein, the binding to the protein, inhibits aggregation of single particles, which of course is useful as well. Aggregation is a big problem with nanoparticles. And this work was published in Current Biology recently. And that's probably the first ever case of absolutely no revisions whatsoever, not even a spelling. She's in Imperial College London now. So proteins involved, use proteomics, the strong affinity, important role. And so the fungi are influencing biomineral formation through protein secretion. And this is kind of relevant to biologically controlled biomineralization I spoke to before, because all the other, lobsters and crabs and barnacles and teeth, there's always a protein matrix involved somehow. So is this somehow some kind of link with some kind of controlled biomineralization processes? And of course, this kind of approach is also useful for manipulation of development of inorganic-based, protein-based nanomaterials, which again is a massive area in sort of chemical bioengineering. So we're able to add to the mechanisms involved in the formation of fungal nanoparticles and nanominerals, redox reactions I mentioned for selenium, and then interactions with the cell wall and with amino acids and proteins, for example, excretion of amino acids and proteins and intracellular peptides and proteins, for example. Now, the nanoscale imbues special properties, as we know, a high surface area to volume ratio and the catalytic and photovoltaic functions, all kinds of antimicrobial functions. But so what could be some of the applications? And what could be some of the benefits to the organism. This is Chan Wei, also an absolute genius. Soon she'll be a professor in Beijing. She's assistant professor now, and was with me. And one of her sidelines, she had the idea of electrochemical materials. So we're making manganese carbonate just through manganese carbonate deposition, the urea interaction, as I've mentioned. And then if you carbonize the mycelium with the associated carbonate, you'll get a carbon manganese oxide composite. And of course, manganese oxide is a very good electrochemical material. It was used in the batteries. It was the black stuff if you used to take the old batteries apart with the carbon rod in the middle. And so, was this a good electrochemical material? Because there's a constant search for electrochemical materials that contain things to improve, ideas about fuel storage and generation. And so these were tested in lithium-ion batteries and supercapacitors, and there's some pictures there of the biomineralized hyphae before treatment. You can see the deposition of the carbonates all around the hyphae, and then here's the carbonized manganese oxide biomass. You can still see a fibrous nature, and there's some lamellar minerals, which are the oxides. And these are also nanoscale because they're about 20 nanometers thick. Nanomaterials, according to the definitions, have to be nano in one dimension, so they're in that one dimension, the thickness. And you can imagine a massive surface area that, of course, lends itself to electron transfer and diffusion of reactants and things like this. And in fact, this composite showed fantastic activity, actually, excellent capacitance, and also excellent cycling ability. About 90% capacity was retained in a lithium-ion battery after 200 cycles. That means 200 cycles of charge, discharge, charge, discharge. And you all know how irritating your decline in charging capacity is of phones and things like this. And so this was a first demonstration of the synthesis of electrode materials using the fungal biomineralization process. And as you can guess, we got a lot of things about this and patents and some publicity. And this was a very interesting expose of the work in the Scottish Sun, which is kind of well known for accurate reporting. Because Neurospora crassa is sometimes called the red bread mold, so yeah, loaf of bread, and of course urea, urine. So they obviously made this, bread using stale bread and pea. And there you can see, and men of a certain age will admire the confident jet of, which obviously must be a younger man in that particular cartoon. Another catalytic activity, again, this was a sideline of Feischwa, degradation of methyl red. And methyl red is an azo dye, and there's azo dye contamination problems. And the copper carbonate nanoparticles can break down hydrogen peroxide, produce reactive oxygen species, degrade methyl red, as you can see there. And those are just graphs that reflect that picture. I'm really drying up. And obviously the absorbance spectra decreasing, and you can see the concentration of methyl red decreasing in solution with time. So reactions and catalytic activity of these nanoparticles. So now, in the last bit, I need to introduce you to the concept of nanozymes, which, again, is a massive area, but of course, microbiology is perhaps a little part of it now, but that's kind of growing. Now, this kind of arose from, I suppose, physicists and biophysicists, things like this. Most life processes in nature involve enzymes, as you know, but enzymes are inhibited or denatured by non-physiological or adverse conditions. Heat, acid, alkalis, and can lose their function, therefore. But nanozymes are inorganic. They're inorganic nanoparticles that mimic enzyme-like properties in redox reactions, and therefore they have the properties of nanomaterials and also this catalytic enzymic function. And there's many applications being explored now in medicine, chemical engineering, agriculture, environment, for example. Now, they vary in structure and composition. Oxides are important, and of course, as I've mentioned, organisms can produce oxides. I mean, that's common actually throughout prokaryotic and eukaryotic fungi. And most of the reactions mimic those enzymes that you'd be familiar with as microbiologists: oxidase, peroxidase, catalase, and superoxide dismutase. And if you pay attention to peroxidase, so nanozymes and fungi, and what is maybe the benefits to the organisms. So I've just told you that fungi can mediate formation of nanoparticles and nanominerals in elemental forms, or carbonates, phosphates, oxides. And so can these behave as nanozymes? Well, obviously they can be reactive, as I've just shown, but what's the significance of this to the organism and significance to the biosphere? And if the nanozyme reactivity occurs in the natural environment through naturally occurring biominerals, we just haven't noticed this. And so this could be an unnoticed contributor to evolution of the lithosphere and the biosphere. Now, with Guanghui Yu and Tianjin, we're involved in exploring this aspect. And of the minerals that can potentially act as nanozymes, ferrihydrate was a chosen topic. You can see there it's a ferric oxyhydroxide, and it's ubiquitous in the terrestrial environment. It's actually a nanomineral, but it can coalesce or whatever together to form like a big mineral that's, you could buy a lump of it on eBay or something if you want. And the catalytic activity of this ferric hydroxide nanoparticles mimics peroxidase, which is an antioxidant enzyme that removes excess reactive oxygen species, such as superoxide and hydrogen peroxide in biological systems, to combat oxidative stress. Anyway, fungi can break down hematite. They can solubilize hematite and biomineralize it to form ferrihydrate. So we studied this phenomena of hematite degradation to form ferrihydrate nanoparticles using a trichoderma species. And you can see some of the results there. There's the diameter of the nanoparticles. You can see absolutely small diameters, a couple of nanometers, increase in number. And concomitant with this process is solubilization of iron from the hematite, so iron 2 is released. And you can see there some using some very advanced imaging techniques, some of the HiFi and EPS, deposition of the iron minerals. There's some EPS, look at the EPS as well, and some SEM and nanosims imaging of iron. And then some of the bottom ones, you can see some of the EPS minerals here, and there's some of the diffraction analyses of these, and there's some of the ferrihydrate particles. You can see really small. The biomarkers there are 5 nanometers. And some of the evidence for the enzymic activity. You can see A and B show location of hydrogen peroxide, blue, and then the brown in A is the oxygen radical here, shown by various staining. And the peroxidase activity of these particles is there, and it's comparable to activity mediated by horseradish peroxidase, using a commercial peroxidase, if you like, in a test tube, which you can see there. So this suggests that these fungal nanoparticles could play a role in regulation of oxidative stress to the organism. And in fact, so a model was made of this, you can see, and this kind of sheath of the biominerals. Iron is released, Fe2, so it ensures iron acquisition. There's the effects of the oxygen radicals and production of hydroxyl radicals, which are actually involved in breakdown of many organic substrates, including pollutants, so that could have a benefit for the organism as well. And so it can mitigate cytotoxicity and ensure essential iron 2 acquisition. And there were some quite novel mineralogical changes and effects in this. Now, non-lattice oxygen was involved, and I won't go into that in too much detail, but suffice to say that this was unusual, and it was an unusual aspect of what was regarded as conventional iron chemistry. And then further, in more recent, there's a couple more papers just coming out now. Again, novel aspects of mineralogy and electron transfer, and also where there's kind of hypothesis that this biomineralized sheath can be a cytoprotective exoskeleton, if you like. I mean, an exoskeleton, in a crab or a beetle or something like this, but it's a bit different. And of course, a lot might be suspended in EPS. But anyway, you'll get the idea. And also, Gang Hui, he likes to refer as if it's spores, but spore coats are tougher. But you know what I mean? A kind of resistant, protective outside, and then the reactive components from the biomineralization process, especially the hydroxyl radicals, involved in breakdown of pollutants and all kinds of things, and also release of essential elements from organic materials. So it looks like there's a lot of importance here and some benefits, perhaps even incidental to the organism. And so it looks like this kind of process may play a more important role in biogeochemical cycling of carbon, nitrogen, phosphorus, and iron than has been previously appreciated, because usually you just think of fungi as carbon metabolism and extracellular enzymes. Oh, for some reason something's moved. Oh yeah, I see the reference there. Now, if you consider that there's teragram quantities of mineral nanoparticles in the Earth system, I mean, they are everywhere, and naturally they're not just, as you might get the impression from Prince Charles or something, that there's somehow that humans have shoveling out. They're all around us anyway. And so it's statistically probable that some of them, if not many of them, will have behavior similar to mimetic catalysts, i.e. Artificial enzymes, if you like. And regardless of the origin of the nanoparticles, over 100 types of natural and engineered nanominerals have been reported to possess enzyme-like activities. So therefore, this work is really describing an unknown route of microbe-mineral coevolution, which raises new questions about the roles of such materials in the lithosphere and biosphere, because obviously if these, through their activities, they're making these minerals that can act as enzymes of benefit to the organism, and it's obviously changing the geochemical and mineralogical components of that environment and going together, and so you can see the obvious link there between biosphere and lithosphere processes and evolution, and further work will be no doubt proceeding to further establish and clarify this. And so to finish off, there's just some simple summary of fungal biomineralization, elemental pools there, precipitation or solubilization, the environmental pools, etc., and the various biominerals, several I haven't been able to discuss in detail here for time reasons, obviously. Applications, bioremediation, biorecovery, products, nanoproducts, soil and concrete stabilization, I haven't mentioned that, but bacteria as well as fungi, you can use them to make carbonate and block poured materials, make protective coatings and things like this. And there are companies doing that. Bioprotection as well, I just mentioned. Benefit to organism, well, there's toxic metal detoxification, there's the benefits that I've just alluded to, iron acquisition and cytoprotection, reactive oxygen species, pollutant degradation. Oxalates are a potential carbon source for other organisms, particularly fungi, and also oxalate-degrading bacteria, stress protection. And environmental consequences, component of biogeochemical cycles, as hopefully you can clear of pretty much all elements. Pretty much all elements have some aspect of biology in their biogeochemical cycle. There's always microbiology in an elemental cycle somehow. And contribution to environmental pool of minerals, metals, metal complexes, etc., patinas, crusts, and rinds and rocks and minerals, biodeterioration of rocks, minerals, and construction materials, cultural heritage, which again I haven't been able to cover, but you can look up. I've got a paper in Nature Microbiology from 2017, which covers all that in the built environment. And wherever you go, you can see. If you look outside this building, you'll see bugs and biofilms and everywhere. As soon as a building's made, bugs are on it and start working. And CO2 fixation in oxalates and carbonates. So there's quite a bit of interest in this. Now, just some of the people, and of course, a lot of this work is really, I've been, 40 odd years, but some is the last few years, and really as I approach the end, at least of my practical research career in microbiology, the last few years that coincided with one of the best groups I've ever had over the 40 years. And I'll say a special for Marina Fomina, who's was with me for many years. She's a senior academic. She's from Kiev in Ukraine, obviously, and many projects. And she's spent the last, few times, as you can imagine, sellotaping windows in her flat, sharing the basement with her neighbours and friends and family and mother. And she's now safely managed to go to a village in West Ukraine with her husband's sister. Her husband's also a scientist, and spearheaded a lot of things. And even when she's in the basement, she's got email. We've got a paper coming out on deterioration in St. Sophia's Cathedral in Kiev, and it's often, this kind of, Ukrainian-y, orthodoxy-looking cathedral blue, very beautiful, and it's a national icon of Ukraine. And anyway, we've got a paper coming out, and we're discussing the revisions, even when she was telling me about the bombs and things. And it shows what a fantastic person she is, strong and a great microbiologist, and hopefully that will come out. And we're going to have a dedication to Ukraine in that paper when it comes out. So many other, Chan Wei, absolute genius, assistant professor, Xinjin, she's actually working in Belfast at the moment, in Queen's University. Fei Shua, she's doing imperial carbonates and selenium and phosphates, all kinds of things, Chan Wei. John Ferrier, a lot of stuff with cobalt bioleaching, and young Junri from South Korea, a lot of stuff on the lead and paramorphite, of course, from South Korea, always did things Gangnam style, so there you go, and that already sounds a bit dated. And Xia, who's now his job in Wuhan. Yui, who's now he's a professor in Wuhan, Chinese Academy of Sciences, and extremely productive. And Lin, or a real name, Bongkutrat from Thailand, and Jayu, who worked on some concrete carbonate precipitations, and Martin, who, again, with me a long time, started literally almost as a schoolboy and ended up being the electron microscope technician. He actually came for a school project once, so you can see sometimes these lab things that you do can lead to good things, and they're all fantastic human beings as well, which makes a difference. Gang Hui in Tianjin, he's an absolute genius. Very happy to be involved with that group with the nanopositive ferrihydrate, and he knows all about iron redox chemistry and things like this. And there's Chan Wei again, who will be in a couple of years a professor, and we'll sort them out in Beijing. And that's her husband, actually, who's a fantastic electrochemical, his job's in Peking University. Finally, just thank my sponsors, Ian Stansfield and Al Brown. I have got some quite, what should we say, alternative photographs of them, but I thought I'd better be respectable and put up fairly normal ones. And okay, so again, thank you to the SGM, the MS, and it's been an honor and a pleasure. So thank you.
I'm sure you'll all agree that was a fantastic lecture by Jeff. I've really enjoyed that. And I'm delighted to present now the Marjory Stephenson Prize Lecture Certificate to Jeff.